Okay, so in this next part, we're going to talk about how the immune system kills. There are a few cells that are very important with respect to different pathogens and eliminating them from your body. And so we're going to talk about them in more detail. We're not going to focus so much on phagocytic cells because we've already talked about how they kill. And so we're going to talk about different cells. So what we're interested in here is talking about more parasitic killing as well as virus-infected cells killing. Our macrophages were important for bacteria, and we discussed that prior. And so here we're going to focus on our mast cells, our natural killer cells, and also our CD8 T cells, of course. So 1/3 of the world population is infested with worm. They're colonized with worms, and this is due to unsanitary conditions. In the developed world, worm infections and parasitic infections are largely not heard of because of our sanitation. However, for the rest of the world, it is a large population that is infected. So one thing to keep in mind with worms is they lay eggs, and the eggs are resistant to acid. And so that means they can pass through your stomach. Now, the major defense against worms are going to be mast cells and eosinophils. Eosinophils have a protein called major basic protein. And this is going to be critical. Major basic protein is going to basically be able to help to digest the outer skin or membranes on the worm. We also have defense against the worm is an increase in mucus secretion. This is similar to an allergic response because allergic response is on the same Th2 pathway as a worm response, and an increase in smooth muscle contraction. And this is used to expel the worms. Your natural killer cells and CD8 T cells are going to be important for virus-infected cells, so these are intracellular, whereas worms are extracellular and large. And so your intracellular pathogens require the cell to be killed, and so that is the job of your CD8 T cells as well as natural killer cells, your NK cells. And they're going to do this both in the same mechanism using similar or actually identical processes, and they all end up undergoing apoptosis. So very quickly, different pathogens, different responses. That's why we have Th1, Th2, Th17, etc. The pathogens are going to live inside of us differently. So our extracellular pathogens, these live in between our cells, in the extracellular space, and they are going to digest your cells to get nutrients. And so extracellular bacteria are going to be important for neutrophils and complement macrophages. These are going to be more Th17 helper cells, IgG and IgM. Th17 because Th17 is less inflammatory than Th1. And when you're in an extracellular environment, you have the ability not only to kill the bacteria, but also to do a lot of damage to your own cells. And so you kind of want to control the inflammation, but at the same time allow your cells, especially your innate cells, neutrophils, macrophages, to be able to ingest and kill those extracellular pathogens. Examples of extracellular pathogens are going to be our normal flora bacteria, our staph and our streps, and also our normal flora fungus infection or fungi, candida. We also have intracellular pathogens. They can be Mycobacterium tuberculosis, but viruses is the most common. The number one cause of infection is virus. Okay, so that's why our Th1 response is so dominant in us, because our pathogens are typically viral and they're intracellular. We can also have mycobacterium infections. Protozoas are intracellular. There are other intracellular bacteria. Listeria can be intracellular. These are just examples. And so our innate response primarily for viruses is going to be interferon. We also have our natural killer cells, which we're going to talk about. This typically has to always be a Th1 response. There has been studies on people who die from intracellular pathogens, and not often, but sometimes it's because they're skewed to a Th2 response, which doesn't protect them, and they die because you need that Th1 response. You need that CTL, you need those NK cells, etc. Our surface pathogens are going to be our hemolyths, worm infections, arthropods. These are going to require the mast cells, eosinophils, and the Th2 response. The Th1 response is not effective. CD8 T cells cannot see anything extracellularly. Neither can NK cells. And so these have to be driven by a Th2 response. Okay. So some things to keep in mind when we start talking about pathogens is we always have to talk about certain disease states. So if we have a defect in our CD4 response, this is going to impair all responses, whether it's Th1, Th2, Th17, Treg, and this is because you have no IL-2 and no help. If you have a defect in antibody, this is going to impair in extracellular bacteria. If you have a defect in CD8, this is going to impair intracellular. If you have a defect in phagocytosis, this is going to impair intra and extracellular. If you have a defect in eosinophils or mast cells, this is going to lead to worm infections. Let's just get rid of that. This doesn't really apply. Okay, so you can see that, and if you think about how we've talked about how T cells develop, how B cells develop, hematopoiesis, any step along all of those lines can lead to impairments, and you can be susceptible to extracellular, intracellular, or parasitic infections. And so here we have an extracellular bacteria, fungi, and protozoa, and these are going to be targeted, of course, by complement antibodies and phagocytosis. And so any defect in any of these are going to impair the ability of your body to respond to those extracellular pathogens. We have intracellular bacteria, viruses, fungi, and protozoa, which require CD8 T cells, and CD8 T cells and NK cells and phagocytic cells. And we also have our parasites that require mast and eosinophils. But important to all of them are the CD4 T cells. They are central to it all, those helper cells. And so you really need those helper cells to help you navigate which type of response you need and to make sure that that type of response is actually induced. So here we have a T helper 2 cell, and this is a parasitic worm infection. And so this T helper 2 cell will release IL-4 because that's a T helper 2 cytokine. And this will induce the B cell, give the B cell help and a heads up that you need to have IgE produced. And the IgE is going to bind to mast cells. And then the mast cell is going to become activated. and release granules. And this is going to kill the worm. One of the things to point out here, because we're talking about this process, is IgE is very unique. It can bind to its receptor without antigen. We talked about IgG being an opsonin. IgG can bind to an antigen and then bind to the Fc gamma receptor on the surface of a macrophage, and the macrophage will eat the IgG with the whatever's attached, bacteria, virus, whatever. IgE is different. IgE gets produced by the B cell in the secondary lymphoid tissue or by the plasma cell in the bone marrow. IgE will go into the bloodstream, will get into tissues where you have tissue-resident mast cells, and the IgE will bind to the surface of the mast cell before the antigen binds. So the mast cell, in essence, becomes primed with all the different IgEs that are made in your body, and it's waiting for the antigen to come in order to get activated. And that's very different than all the other antibodies. Okay, so let's talk about mast cells. So again, mast cells are unique. They are going to be similar to a macrophage in that they are tissue resident. They are going to have on their surface IgE receptors. And these IgE receptors are going to bind IgE. So we can, for example, have peanut IgE. We can have shellfish IgE. We can have grass IgE. We can have pollen IgE. And they'll all be bound to the receptor, which is an Fc epsilon for E, receptor 1. And all these different IgEs with different specificities will bind to the same mast cell. So you can think of the mast cell sort of like a pincushion with different colored pins that respond to different antigens. So keep in mind, the mast cell in the bone marrow, and it's going to be driven by IL-3 and IL-4 to become a mast cell. And then it's going to go to a tissue, and it's going to bind IgE, and then it's going to wait. It sits there and waits. It waits until it's grass season, and you take in some grass, and then it says, oh, I have grass IgE, I'm going to release all my granules, and you're going to have an allergic response. The IgE on the surface and the Fc epsilon receptor 1 on the surface of your mast cell gets activated through cross-linking. And cross-linking, 1 requires antigen, 2 requires IgE, and 3 requires Fc epsilon R1. And so here you have, here you have your antigen, those are yellow dots binding to IgE on the surface, and that is called cross-linking when you have multiple dots binding to multiple IgEs on the surface of a single mast cell. And they're going to cross-link, and your mast cell will then degranulate. Okay, so a few notes though. C5a, complement 5a, or C3a can also activate mast cells. But that would be more of an innate type of activation rather than a specific antigen type of activation. Activation of mast cell and degranulation of mast cell is referred to as immediate hypersensitivity. And it's because it happens quickly, You have sensitization with antigen, and it goes to a Th2, which drives an IgE response. And then you have antigen exposure, which drives the release of granules. So this is a primary response, and this is considered a secondary response. So we talked about activation to effector to memory, and then memory cells occur faster, and that's the same thing with this mast cell story. Is the first time you see the antigen and you respond to the antigen and make IgE, you don't get an allergic response. You don't get an inflammatory response. It's the second time you see that antigen is when your body gets that secondary response, that allergic response. So mast cells also have different types of mediators. So it has preformed mediators, and that's why it's called immediate hypersensitivity, because these immediate preformed mediators get released almost immediately. So it's within seconds. And then the late mediators are going to occur within minutes to hours. So with respect to the immediate hypersensitivity, which is the allergic response, the difference between immediate and late is not days and weeks, but it's just hours as opposed to minutes. Okay, so we have our immediate response, or IR, is seconds, and we have our late response, which is minutes to hours. Okay, so in our immediate or preformed, we have some of the, and this is immediate response, we have some of the more well-known mast mediators. We have our histamine, which people who have allergic responses know histamine as an immediate response. And histamine is going to cause smooth muscle contraction in many different tissues. And it also is going to increase vascular permeability. And in essence, it evolved this way because it wants to get cells, especially eosinophils, to the site of where this parasitic infection is. And so you're going to get endothelial cell contraction and widening of gaps between cells, which allows white blood cells to enter. This also leads to an itchiness. Now we also have proteolytic enzymes. Tryptase and chymotrypsin are going to be released. And these are going to activate complement and the kinin pathways, kininogen and bradykinin. They increase mucus secretion. They are going to lead to smooth muscle contraction in the bronchi. And they're going to activate the complement and kinin pathways. Tryptase is actually going to be released in the blood levels, and it is going to be a measurement of serious allergic responses. Now, there are some also immediate reactions with respect to your prostaglandins. So your prostaglandins are going to be released very early. They're going to cause vasodilation, vascular permeability, constriction of smooth muscles in the gut and bronchi. And these are going to act within seconds. And of course, you know that these are made by cyclin oxygenase. Now, our late response is going to be things like our cytokines and our leukotrienes. Our cytokines are going to be Th2 skewing. So they're going to help to keep promoting a Th2 response to these particular antigens. They're going to activate your endothelium, enhance diapedesis. That's what TNF does. It's one of those acute phase response proteins. Your IL-4 is obviously a Th2 driving cytokine. And then IL-3 and IL-5 are going to stimulate eosinophil production, release, and activation. And so again, this goes back historically to eosinophils are going to be a major defense mechanism against parasitic infections. So here we have, as a reminder of our prostaglandin and leukotrienes, we have arachidonic acid. And so mast cells and eosinophils can generate from arachidonic acid different mediators. So using the cyclin oxygenase pathway, you can have prostaglandins as well as thromboxane. Using the lipoxygenase pathway, you can get your leukotrienes as well as your platelet activating factor. So your prostaglandins are going to inhibit Th1 responses. Your leukotrienes and thromboxanes are going to be for vasodilation, smooth muscle contraction. Platelet activating factor is going to be chemotactic and is important for activating eosinophils as well as your neutrophils. So let's talk a little bit about eosinophils. Eosinophils are a granulocyte. They must be recruited. Their granules are very toxic, so you want to control them. They have very low numbers in the blood unless you live in a parasitic endemic region. You have IL-3 and IL-5, which will increase their numbers. And this equals eosinophilia. There's also eotaxin. And this is going to recruit the eosinophils to the site of parasitic infection. And leukotrienes can do the same. So eotaxin is going to be made by epithelial cells, and leukotrienes, of course, by our mast cells. Sorry, spelled. Now, we have here our eosinophil, and the eosinophil is going to, the granules are stained pink. So let's change to pink. And that means they have, they're binding or they're staining with acidic dye. And so what this means, therefore, their proteins are basic. And so you have a few very important proteins or important functions of the eosinophil. So the first is you have peroxidase. And peroxidase will sit on the parasite's body. and it will form hypochlorous acid. And this, of course, will eat away at the parasite. It also has major basic protein, which also eats away at the surface of your parasite. And it has cationic protein. I'll put it here. And this damages the surface. Plus, it's a neurotoxin for simple nervous systems. Okay, so just a moment, we'll talk a little bit about eosinophilia and Schistosoma mansoni. This is a common parasite. It is found, the larvae are free swimming and burrow into a swimmer. So it's found in water where you have the plant. It's a hyacinth plant, and it's snail host, which feeds on the hyacinth plant. And then the snail is part of the life cycle of the parasite, and it releases free-swimming larvae. And of course, the free-swimming larvae want to burrow into more snails, but if a human is swimming, they will burrow into the humans. And the eggs are found in the liver. It causes inflammation of the liver, and it can lead to cirrhosis of the liver if untreated. Okay, so what we've been talking about here is type 1 hypersensitivity, which is immediate, and this has to do with mast cell degranulation. And this is within seconds or minutes. We also have the late phase, where you have cytokines, and these can lead to a chronic allergic response. In the developed world, the leading cause of eosinophilia is going to be allergies. In the developing, this is going to be caused by parasites. Okay, so we're moving away from mast cells and eosinophils and how they kill with their granules, and we're moving more towards cell-to-cell or cell-mediated killing, and that's the next topic. And so we're going to talk about natural killer cells. So some things to remember about natural killer cells is you have a macrophage that releases IL-12, and this activates our NK cell, which then releases interferon gamma, and this makes the Th1 environment. So that's really what a CD4 T cell needs to see in order to become a Th1 helper cell. So one thing to keep in mind about natural killer cells is they can target cells plus kill cells without prior activation. Or sensitization. They are, of course, innate. No specificity. So they're able to kill cells. T cells have to be activated. Mast cells have to be sensitized with IgE. Natural killer cells need neither. So what is the natural killer cell really doing? Well, a natural killer cell is going to, similar to our innate response that looked at pattern recognition receptors, the natural killer cell is going to look at a pattern on our cells. It's not looking for a pathogen marker, it's looking for a cellular marker. And it's a cellular marker for health. So the one way to think about this is the natural killer cell, every nucleated cell in your body expresses MHC class 1. And your natural killer cell can recognize MHC class 1. Oops. And if it sees MHC1, then the cell lives. If it does not see MHC1, then the cell dies. And this is via apoptosis. Okay, so in essence, a natural killer cell will kill cells that downregulate MHC. So and this can be caused by viruses. We have herpes viruses prevents MHC1. And so your CD8 T cell won't be able to see it. So in this picture here, we have an infection with flu that does not downregulate MHC1. And so your natural killer cell will not kill a flu-infected cell because there's plenty of MHC1. But a CD8 T cell will kill a flu cell because it recognizes the flu antigen associated with that MHC1. Herpes simplex virus downregulates MHC1. And so herpes simplex virus will not be seen by a CD8 T cell because there's no MHC1 on the surface. However, the herpes simplex virus, because it downregulates MHC1, the natural killer cell does not see MHC1, and so it decides, I need to kill this cell. So there's two scenarios where a potential virus-infected cell can be killed. One is by a CD8 T cell recognizing antigen on the surface of the infected cell and killing the infected cell. The second way is for the natural killer cell to not see MHC class 1 on the surface of the infected cell and killing that infected cell. So let's talk a little bit more about how your natural killer cell does this. So again, you have a virus-infected cell. And you have two outcomes. One, if MHC1 is present, the cell survives. And two, if MHC1 is not present, then the cell is killed. Now, one thing to keep in mind is that it's not as easy as this. It's actually a very elaborate and very controlled process. So there are about 20 different receptors that activate or inhibit NK cells. They are a very fluid population and a very complicated population. Th1 cytokines IFN gamma increases MHC expression. And this is important because pathogens that are intracellular are going to induce a Th1 response. You're going to get interferon gamma, and it should increase MHC expression on the surface because you want the CD8 T cell to be able to find the infected cell. Interferon gamma also increases NK cell activity. And this is because there are many viruses with virulence factors that will interfere with MHC1 expression. And so you need the NK cells to be active so that they can go around and find those cells that are not expressing MHC. On the NK cells, you have something called KIRs. And KIRs are killer Ig-like receptor. So that's what a KIR is. And then NK cells are also going to be involved in another process called antibody-dependent cellular cytotoxicity, which is at ADCC. But I first want to talk about KIRs. So a KIR recognizes MHC1 alpha chain, not the region that binds to the peptide, but the region that is for the structure of the alpha chain. We have NKG2 and CD94. That recognize HLA-E. And both of these will inhibit killing. So if either of these are engaged on a natural killer cell, even if the cell that they're looking at is infected, they will not kill that cell. So what this means is inhibitory cells dominate. Or signals, sorry, not cells. Inhibitory signals dominate. Now there is also NK activating receptors. NKs have an FC, again, we're talking about antibodies, FC region, gamma, so we're talking about IgG, receptor 3. And this recognizes the FC region of IgG bound to the cell surface. And this triggers killing. And this is referred to as antibody dependent cellular cytotoxicity. What is that? ADCC. Keep in mind that macrophages also have FC gamma R3, but this is for opsinin only. It has nothing to do with killing. And also your placental NK cells, not CD8 T cells, are involved in protecting the fetus from infection. Okay, so how do T cells and NK cells kill? Well, they both kill in the same way. They're both going to induce apoptosis. So that's straightforward. So programmed cell death. So keep in mind, an activated NK cell or CD8 T cell increase fast ligand. And fast ligand is one of those ligands that have that can trigger a death signal. So we have here a few ways that T cells and NK cells kill. I'm going to talk about one way, which is not shown in this picture, and this is by pre-made proteins in granules. And you have porphyrin, which polymerizes and forms pores, sort of punches holes in the target cell. And then you have granzyme, which are 3 separate enzymes. But the important thing to know is that they are proteolytic, so they degrade proteins as well as activate caspases. Now, a second way to kill is through that fast ligand. So fast ligand is going to be expressed on the NK cell or your CD8 T cell. Your fast is expressed by some cell that's stressed or infected or needs to die. The fast has a death domain in its cytoplasmic tail, and this will initiate apoptosis. And then the third way of killing is through TNF. So your natural killer cell, your CD8 T cell, or a macrophage will release TNF either free or membrane bound. It will bind to the TNF receptor that also has a death domain on the cytoplasmic tail, and this can lead to apoptosis or another change within the cell. So FAS plus your TNF receptor have death domains. And these are going to activate caspases. Now, caspases are just simply enzymes that cleave proteins, and they target aspartic residues. They activate the DNA that cleaves the DNA into 200 base pair fragments. They disrupt the mitochondria and make it leaky. Now, what this is showing here is you have two sort of opposing signals going on in this cell. On the one side, you have IL-2, and this is giving a signal to survive through BCL2. And then on the other side, you have FAS, and this is giving a signal for death through caspases. And so BCL2, as long as IL-2 is present, will protect the cell from undergoing apoptosis. But as soon as IL-2 levels go down, the cell becomes susceptible to apoptosis because it's lost that anti-apoptotic BCL2 signal. BCL2 stabilizes the mitochondria. And memory cells have high levels of BCL2. So they survive longer. So let's just talk a little bit about apoptosis versus necrosis. So apoptosis is programmed cell death, or necrosis is inadvertent cell death. It wasn't meant to happen. The cell didn't want to die. The causes are going to be during development. So apoptosis is normally used during development. It's also used normally for the contraction of immune response. The T cell and K cell killing that we just discussed, etc. In programmed cell death, the DNA will break down, but the cell remains intact until the phagocytic cell comes along because of phosphatidylserine on the surface and eats the cell. There is no inflammation. It's a quiet type of death, and usually it has no adverse effects to the host. Necrosis, on the other hand, is usually caused by some sort of damage. It could be heat, chemical, or actually an infection. There are some viruses, for example, that are cytolytic, which means they cause cells to explode. The cell membrane will break down and leak its contents, and it does cause inflammation primarily due to activation of complement. And it usually has no positive effect on the host. Now, just as a side with respect to apoptosis, apoptosis is used just normally to control things. During negative selection in the thymus, peripheral T cell tolerance, your T cells that are potentially autoreactive are going to undergo apoptosis. During contraction of immune response, which has to happen because your cells have to go back to a normal number, the responding T cells, or most of them, will not be able to compete for survival signals, and they're going to mostly undergo apoptosis. Negative selection of B cells, again, this is a tolerance mechanism in the bone marrow. We have B cells that are going through clonal expansion in the germinal center reaction, and if they can't compete and they are low affinity, they don't bind very well, they undergo apoptosis. We mentioned already peripheral T cell tolerance, privileged sites. You have your endothelial cells at privileged sites expressing Fas ligand, so that if any immune cells come in, they get deleted pretty quickly. Intracellular infection, and this is through your NK cells and via NK, mostly CD8 T cells. So even though NK cells can target virus-infected cells that downregulate MHC, most viruses do not do this. And so it's mostly going to be the CD8 T cells that are going to be protective against an intracellular infection. Malignancy, again, can have the same thing. You can have a T cell that targets it, or downregulation of MHC, and NK cells targets it. And defects in apoptosis, remember, defects. Right, sorry. Can lead to malignancy. Okay, and thank you